Hello, welcome to the next installment in the biology series. So we've got a very complicated diagram here. What we're going to talk about is metabolism, how our metabolisms work. There are a few quite complex actions within this metabolism that I'm showing here, but we'll go into more depth in those things like the Krebs cycle, like oxidative phosphorylation a bit later. But what I want you to know is how the metabolism works and how it makes use of food, basically, and all the macronutrients in food. So you can see here the macronutrients. We've got the carbohydrates, we've got the fats, which are triglycerides, and the proteins, which is what we eat. Each one of these macronutrients then goes through the digestive system. Here we eat it in, and it goes all the way through the gastrointestinal tract, and eventually makes its way into each individual system. So the carbohydrates make their way into the blood, aiming for the liver. The triglycerides, the fats, make their way into the lymphatic system, the lymphatics. The protein also makes its way into the blood. So let's talk about what happens when it gets there. So the carbohydrates makes its way into the bloodstream, and the micronutrient of carbohydrates is glucose, which is a thing we shuttle around our body. So once you eat carbohydrates, all types of carbohydrates, they will turn into glucose. And glucose then can do two things. It can go through the system, which we'll talk about, or it can be stored into glycogen. This process of storing it into glycogen is called glycogenesis. Also, we can obviously turn glycogen back in to glucose to use it once it's from its stored form back into glucose. That is glycogenolysis. But what we're really looking for is glucose. That's what we want to use. So once we've stored it into glycogen, then we can unstore it and use it again as glucose. Now, glucose then goes through the process of glycolysis. And the glycolysis is a few long steps, not just this one here that I've got. There are a few steps in here, but really the idea of glycolysis is to get to pyruvate through a number of steps, one step being glucose 6 turned into glucose 6-phosphate, and then into pyruvate. The whole reason why glucose turns into pyruvate is because glucose cannot go through the phospholipid bilayer, which we've talked about before in the previous episodes. So if you want to know more about that, then go take a look. But it turns into pyruvate. Pyruvate is an easier molecule to get through that phospholipid bilayer and into the mitochondria, which is what we have here. This is the mitochondria with its double layer membrane. This is what we're after. Glycolysis and all these things take place outside of the mitochondria, in the hepatocyte, the cells of the liver generally, but then enters into the mitochondria to perform the Krebs cycle. Now, once the pyruvate has entered into the mitochondria, it then turns into something called acetyl-CoA. And acetyl-CoA, using its friend oxaloacetate, join together to start off the Krebs cycle. And this cycle, the tricarbolic cycle or the citric acid cycle, Krebs cycle, all the same thing. Goes through a number of chemical exchanges. And the whole idea of this cycle, throughout all of its chemical exchanges, is to get the chemical forms of NADH and FADH2. What these do, then they can release electrons and hydrogens into the electron transport chain, which is on the membrane. These electrons enter into 1, 2, 3, 4 complexes in the electron transport chain, and the hydrogen follow. And the hydrogen are then pushed from the matrix outside outside of the membrane, causing a low concentration gradient of hydrogen on the inside of the mitochondria. That then brings into play something which is called the ATP synthase, which is this channel here, this protein channel inside the membrane. What happens then is that gathers up all the hydrogen, turns it into ATP, a vast amount of ATP. And this is the currency, ATP, that we use for energy, which is then distributed all the way around our body. So you can see that the Krebs cycle basically just does chemical exchanges, aiming to get as many FADH and FADH2 molecules as possible, throws it then into the electron transport chain, which then uses the hydrogens through the ATP synthase to make ATP, which then goes around the body for energy. And we'll cover these two, the oxidative phosphorylation, which is the electron transport chain, and the Krebs cycle. We'll cover those in more in-depth look in further episodes, but this is the basics of what is happening here. But there's one other thing on this side of the diagram to touch on, is the lactate. I should have said that all of this happens in the presence of oxygen. Oxygen is needed for all the chemical bond breaks, all of the oxidative phosphorylation. Everything here requires the oxygen to work its magic, breaking down the chemicals. What happens if we don't have oxygen then? And what happens is everything starts to back up. It backs up, backs up, backs up, backs up, backs up, backs up through the system, backs up, backs up, backs up, backs up all the way back to pyruvate. And then pyruvate turns into lactate. Lactate will then gather up all the free radical hydrogen, wait out until the oxygen's ready, and then pump it straight back into pyruvate, and we're off again. Okay, so we need the oxygen to get going. Yes, we can go for a little while utilizing lactate without oxygen. And I must stress that's not lactate, lactic acid, like we used to think, that once oxygen backed up, it backed up into lactic acid. No, that's no longer the case. And we don't believe that happens anymore. Now turns into lactate. Lactate gathers up all the hydrogen and then waits out for the oxygen, which is obviously not long. You can't do that process for very long. As soon as the oxygen is present, then jumps back into pyruvate, back into the citric acid cycle. Start over again. Okay, so that's that covered. Now, what about if we don't have carbohydrates? What about if none of this process on this side can happen? Well, you say none of it. Quite a lot of it will happen without carbohydrates. In fact, the triglycerides that we eat, fat, basically goes into the lymphatic system. And then that is broken down into fatty acids and glycerol. So you have the triglycerides, which are 3 fatty acids and a glycerol backbone. Now the glycerol can be used, turned into glucose 6-phosphate, and then into pyruvate, and we're away again. Same process, everything going through the same way it does as if we was eating carbohydrates. So that all works very, very similar. The fatty acids from the breaking down of the triglycerides through the lymphatic system jump straight into acetyl-CoA. So we have 3 fatty acids there that can jump straight into the the Krebs cycle. Jump straight into the Krebs cycle, continue with the Krebs cycle, citric acid cycle, whatever you want to call it, producing the FADH2 and the NADH straight in to the electron transport chain, ATP. Hello, here I am. So there we go. But what about protein? Okay, protein is essentially broken down in our body to amino acids. Yes, some amino acids are sent to the kidneys to end up as urea. But other ones like alanine, which are great amino acids, they can be thrown straight in to the Krebs cycle and used as energy. And also some amino acids undergo glycolysis, jump into glucose 6-phosphate, turn into pyruvate, and away we go again. So fats and proteins kind of already have energy covered. But there is something else. When the body cannot produce enough pyruvate, we begin to lose energy. The cells realize that energy is of paramount importance. So it needs to have this pyruvate to turn into acetyl-CoA. Now, oxaloacetate in the Krebs cycle cannot just turn into acetyl-CoA. These two are kind of like brothers. They need to work together to set off the Krebs cycle. So we need oxaloacetate and acetyl-CoA to be separate molecules. But if we're not producing enough pyruvate through energy, through glycerol, through glycolysis of amino acids, then we're not going to be able to get enough acetyl-CoA to help oxaloacetate perform the citric acid cycle. So what happens? Well, that's what this arrow here is for. Okay, oxaloacetate can actually turn itself into pyruvate through, again, a number of steps. I'm very, very simplifying this, but the aim of it is to turn itself back in through the membrane to pyruvate, which can then turn itself through chemical exchanges back in to acetyl-CoA. So in a way, the oxaloacetate is offering up its services to change itself into pyruvate, to then give pyruvate a chance to make more acetyl-CoA. The problem here is that oxaloacetate then diminishes within the mitochondria, and we end up with more acetyl-CoA. So again, we have the imbalance. Whereas before we had too much oxaloacetate and not enough acetyl-CoA. Now oxaloacetate has jumped out, pyruvate has come in, so we have less oxaloacetate and more acetyl-CoA. What happens here then? We've got too much acetyl-CoA. What happens to it? Well, this is how ketone bodies are made and how you are in ketosis. These acetyl-CoAs stack together like Lego blocks and perform and turn themselves into ketone bodies like beta-hydroxane butyrate, which then goes to the brain, and then the brain, very helpfully for us, turns it straight back into acetyl-CoA. So this little process here is its own little cycle of ketosis. Can we then get energy without carbohydrates? Let's have a look. I've taken out the carbohydrates. Are we still getting energy? Yes, we are. We're getting lots because the glycerol from the fatty acids are turning into glucose 6-phosphate, pyruvate, and the system starts all over. So this whole process here without the carbohydrates is actually called Gluconeogenesis. Gluconeogenesis. Genesis stands for the beginning of, neo, new, and gluco is the precursor to glucose. So it's the beginning of new glucose. And that's what this whole process here is. And our body functions very, very well without any carbohydrates. However, let's imagine you are a top athlete you want as much energy as possible. Your body is very, very good at keeping this process going. Let's pretend you are a fat adapted athlete. You have gone through the process of reducing carbohydrates, and not only reducing carbohydrates, but reducing carbohydrates whilst you train as well. If you want to be a truly fat adapted athlete, then you must train with reduced carbohydrates. But let's imagine you want optimal performance. Optimal performance is going to require every single bit of energy you have and every single bit of energy your body can produce. So whilst this system here, this gluconeogenic system, is great, it can give us as much energy as we need on a daily basis, even for the high intensity exercises. Should you have gone through the correct training procedures with this particular system. However, this is why athletes, especially top athletes, utilize carbohydrates to get the most out of their body. Watch this. Now what's happened is we've managed to get more energy from more resources so that our body can optimize energy. And if you are a top athlete, if you need to make sure you can get energy from everywhere, then utilizing carbohydrates is often a good thing. Do we need carbohydrates? No. Do we need them for absolute optimal performance? Are they beneficial to get the best out of our body? Yes. Should they be consumed on a daily basis? No. So there you go. That is the metabolism for you.